So, if you ever want to be humbled, just watch yourself on video for a few minutes. I took the time to do so, and I was horrified. Don't care a whole lot for my voice. Don't care for some of the mannerisms. You know, I know this is here, but I don't see it. You know, when I look in the mirror, I don't see it, so it doesn't bother me so much. So I'm going to try to look up like this the rest of the day. My dentist has been after me for years to get braces to fix my bottom teeth. They're not that bad, but you know, that's what dentists do. Finally, last time we were with him, I said, Doc, listen, before I pay for teeth, I think I'm going to get hair. So, for those of you that have had to watch it a second time, I'm really sorry. We'll work on making it a little bit better here. So, last time we were talking about the dynamics of prokaryotic growth. Just like to review some of the stuff we had going on. So, if you remember, we introduced our bacterial growth curve. And this was a growth curve that represented the growth of bacteria in the laboratory, in a closed system, where there would be no additional nutrients added and there would be no waste taken away. And so the environment for the microbes changes over time, and it affects their ability to grow. One other thing I would want to point out about this growth curve, it's going to be, for each of the microbes, where they are in the growth curve is going to be dependent on where they are in the colony, if you think about it. So if we had a colony here growing, you can imagine that those microbes that might be in the center of this colony, probably the first microbes to establish the colony, they're probably way over here, maybe even in the death phase of our growth curve. Whereas if you look on the edge of our colony here, you're probably going to find microbes that maybe are more in the logarithmic growth, or maybe they're up here because of accumulation and stuff. But they're probably in a different area in the growth curve, depending on where they are in the colony. That is not going to be the case when we consider cells that are growing in a culture now. Cells growing in a culture are probably all in the same place with regards to the growth curve, because the depletion of nutrients and the accumulation of waste are pretty much going to be evenly distributed for the most part in a broth culture. Of course, things can't settle, and so that can affect the distribution. But generally, we would assume that cells within the broth culture are pretty much in the same phase of the bacterial growth curve. These guys could be at various phases within that growth curve. We introduced environmental factors that can influence bacterial growth. We talked about temperature as a factor that will influence growth. We mentioned pH. We mentioned oxygen availability as factors, environmental factors that affect growth. And then we identified various groupings within each of these. I won't go into those, but you'll know those, of course. And water availability. Sorry, I almost left one out. And then we were just getting ready to talk about nutritional factors. That will influence microbial growth. So we talked a little bit about nutritional factors when we were talking about food, but now we're not talking about food. We're talking about things that are in the microbes' environment that they're going to have to get. And of course, it could be growing on food, if that is their environment. But we also got to realize that we're probably talking about microbes that are in the soils, microbes that are in water, microbes that are associated with plants. They all have to get what we would call required elements. Those are things that all life has to have in order to grow and reproduce. So carbon and oxygen and hydrogen and nitrogen. And generally these things are going to come from, you know, the foods, the diets that they're eating. Their carbon source, if you will provide a lot of these required elements. Of course, some microbes will produce their own food. We'll call them phototrophs. Others that have to go out and get their carbon source, heterotrophs. We've already kind of covered those two terms. Now, there are other elements that might be needed as well. So inorganic salts might be something that the microbe needs. And then trace elements. Things that the microbe needs in very small amounts. So copper and zinc and cobalt, manganese. These are things that the microbe might need in small amounts in order to grow. And for the most part, we would expect that a lot of these things will come from the microbe's environment. We should think all of it will come from the microbe's environment if it's growing in that environment, if all other factors are being met. However, some microbes require additional growth factors. When we are preparing media that we need for growing bacteria, we can usually cover most of this stuff by throwing in some beef extract or some yeast extract, some stuff like that generally will have most of these elements. But some elements might not be there. Certain cell constituents that can't be synthesized, for instance, amino acids. You know, there are 20 different amino acids, but maybe the microbe can't make all 20. So the amino acid would have to be in the media in order to grow the microbe. Or certain vitamins maybe would have to be there. These are things that you might have to take into account if you're having difficulty growing a microbe on a media. That maybe there are other growth factors, growth elements that you haven't considered. Microbes that require a lot of different growth factors are said to be fastidious. So when we go to grow bacteria in a laboratory setting, we have to consider what the purpose is, what we're trying to isolate, the types of microbes, the environment that they came from, and trying to determine the media that we want to use in growing and cultivating those microbes. There are different kinds of media. A complex media is one that we would find a bunch of ingredients, a variety of ingredients in it. So like I mentioned before, peptone and yeast extract and other things that would allow for a general bacterial growth to occur, general population of microbes to occur. Like the nutrient agar that we use in laboratory, that's a more general medium that just kind of lets everything grow for the most part. It's not one that would inhibit. Sometimes though, microbes, because they are fastidious, might need a more chemically defined medium, one where we know exactly what is in the medium. We've measured everything, and so we can tell you the pure chemicals that make up the media. If we're throwing in beef extract and yeast extract, you know, this is a compilation of things and not necessarily pure. We might want to use a selective medium if we are looking for a particular microbe and we know that there are other bacteria that might be in the community that we don't want to grow. we might use a medium that will favor the growth of another, inhibiting the other. So one that would be more selective. And then finally, we could also use a differential medium. This one here isn't one that necessarily inhibits the growth of other microbes, but particular microbes will show a certain growth pattern. A lot of times they'll show a color development on the media, and so we can differentiate those particular microbes from others that might be on the plate or might grow on that plate. The example of that was the EMB media that we used that had the E. coli with the metallic green sheen. That's a differential medium. Only the E. coli will produce that metallic green sheen. Other microbes that are growing on there will grow maybe, but they won't show that metallic green sheen. So that differentiates the E. coli from other microbes that might grow on that media. There also may be a need for enrichment cultures. Enrichment cultures will allow us then to hopefully grow and isolate culture microbes that might be in small numbers. Essentially, you might have a situation, imagine now I'll set up 3 tubes here, and initially the microbe I'm interested in, maybe there's only one microbe per mil. If I were to try to streak this onto a plate in hopes of isolating this microbe on a plate, and my chances are probably small because there's so many other bacteria there. So what I want to do is try to enhance the growth of these guys so that I can increase their number, and hopefully then, if I need to, transfer to a plate streak for isolation. So I can transfer to a medium that maybe is more favorable to the microbes that I'm targeting, and so we get increased growth of these microbes here. You might still get some growth of the others, but not as much because of competition now. And then by the third transfer, you might have enriched your culture enough that if you were to streak this on a plate now, you would easily get isolated colonies of the microbe you were targeting. Our LTB, when we ran the multiple tube fermentation test, was an example of an enrichment culture. That will do it for that exercise, or excuse me, that chapter. Any questions with regards to chapter 4? Yes.
You know, in the previous lecture, you said all the eukaryotes and prokaryotes have that flagellum.
Not all, no. All prokaryotes have flagellum. Not all prokaryotes have flagella. So no, they don't all have flagella. But those that have flagella, the flagella are made out of flagella. And that's a uniquely prokaryotic molecule. So eukaryotes won't have it.
What I was trying to ask you was, you know, you are using flagellum, flagella. So we want to know the difference between those three.
This is a molecule that the flagella are made of, okay? Singular, plural, okay? So these are the structures, the whip-like structures, and this is what they're made of, okay? Anything else? Yes?
Which one's plural?
Flagella? Flagellum would be one. Flagella, many. So, moving on to chapter 5, we're going to talk about controlling microbial growth. In the 19th century, if you were to get sick and have to go to the hospital and have surgery, there is a good chance that you might survive the surgery and die from infection afterwards. As many as 50% of the individuals that survived the surgery would die from post-surgical infections. Part of the problem was the hands. The doctors didn't wash their hands between surgeries. One of the issues for women during the time was a condition called puerperal fever. It was an infection of the uterus that happened after giving birth. The doctors would be in surgery with somebody, maybe remove an organ or something, take that organ, maybe stick it in a pocket or a bag or whatever, be done with it, and not wash his hands, and then go to a delivery ward and deliver a child. And so, Streptococcus pyogenes, the microbe that causes this, would infect the uterus, and often, you know, the woman would die from the infection passed on from the doctor. Well, in the 19th century, observing the fact that a lot of patients would come down with post-surgical infections, Joseph Lister instituted a program where he would treat post-surgical wounds with carbolic acid. He would take rags and soak them with carbolic acid and lay them over the post-surgical wound. And it worked in